It's a really nice day today, and I decided I'm going to upload at least 10 videos every day, but I don't know if I'm going to stay at home the entire day. Anyway, welcome back to another video. In the previous video we were talking about nucleic acids and nucleotides, and we were zooming in the DNA and into the RNA as the genetic materials, and we talked about the fact that the DNA and RNA are the genetic instructions, genetic material that gives us the instructions to build proteins from the sequence of amino acids. We still didn't understand how it happens, how the DNA is transcribed into RNA and the RNA is translated into proteins. We are going to do it in the future, but for now I want to actually understand the structure of DNA and RNA. So in the previous video I gave you first of all the condensation reaction between different monomers, the nucleotides, and we saw that the different nucleotides can form a form between the 5' carbon to the 3' carbon hydroxyl group and phosphate group, which causes the ester bonds. The ester bonds are called phosphodiester bonds, and multiple phosphodiester bonds between many nucleotides will give us a DNA strand. The DNA strand will occur from the 5 to the 3 end of the strand. Now these terms are very important to remember. Just remember one, the DNA sequence is from the 5' end to the 3' end. The next step I would like to add and talk about in this video is that even before scientists were able to actually understand what is the structure of DNA, when we had the sort of technology that allowed us only to analyze the form and the components of DNA before actually we were able to see the structure, we saw something pretty interesting and weird. We saw that the ratios between different nucleotides is the same, is equal for different nucleotides. So we saw that guanine were in the same ratio with cytosine in DNA, and we saw that thymine was always in the same ratio with adenine. So if we have 30% of the molecule of the DNA that we analyzed in the lab, 30% of guanine, we also had 30% cytosine. And then, in order to complete to 100%, we have 20% of thymine and 20% of adenine. The ratio between thymine and adenine and guanine and cytosine were always equal and the same. It led to this realization that DNA is probably built from two equal molecules, that come together and combine together. And once we realize how to actually see the structure under the different technology tools we have around 70 years ago, we were able to see that the DNA structure is a double helix. And I'm sure you know the double helix picture from many different movies and from general knowledge. So the following structure is the double helix of the DNA, of our genetic material. An interesting fact I would like to add is that if you take the entire molecule inside each cell that we talked about, we have multiple long molecules of DNAs. If we take the longest one, which is chromosome number 1. Don't worry about it. I know you still don't know about chromosomes, but just know the general fact that if we take the longest DNA molecule, which is chromosome number 1, and we connect it to all the other DNA strands, DNA molecules we have inside our cell, all together, start to end, all the way, if we take the DNA molecules from our entire body, we'll be able to connect the DNA to reach the end of the solar system and back to Earth only from a single person's DNA molecules. This is just to show you how much DNA we have inside our body, which is a pretty nerdy and a nice fact I wanted to add. Anyway, as very similar to the alpha helix structure of the secondary structures of protein, we can see that hydrogen bonds can give us the conformation of the molecule itself. Unlike alpha helix, which is only made from a single strand and every 3.6 amino acids we had the hydrogen bond, in DNA helix we have two parallel, or in our case anti-parallel strands that come near each other and form hydrogen bonds between the nucleotides. So this part is extremely important, and I would like you to wait a second and follow me while I'm explaining it, because it's so crucial and it's the basic of DNA and the molecular genetics. So we understood lately, after a few years of research, that guanine and cytosine can come in the same ratio because they can form hydrogen bonds together. And cytosine and guanine can form 3 hydrogen bonds together. On the other hand, thymine and adenine can form 2 hydrogen bonds together. The following conformation and formation of hydrogen bond, the following structure is stable, and the following 2 and 3 hydrogen bonds, because of their connection and orientation in space, it gives us the helical shape of the DNA, the double helix. Because of the different length between the double bonds, the amount of double bonds, the size of the nucleotides, it will give us overall the shape of the double helix, which is very important to remember. So what determines the fact that DNA is helical? The fact that we have 2 strands, one come from the 5 to the 3' end, and the other one is anti-parallel. It comes to the 5 at the bottom, parallel to the 3 at the other side, to the 3' end, and the molecules, the nucleotides, all form double bonds, and they form hydrogen bonds together among each other. Guanine can only form triple bond with cytosine, and thymine and adenine can only form double bond between each other. You won't see thymine to cytosine or guanine to adenine because thermodynamically it is not a stable state, and the DNA helix will collapse or will be deformed. Therefore, the more order thermodynamically state to reach is where you have hydrogen bonds between guanine and cytosine, and thymine and adenine. It's a very important thing to remember. The following hydrogen bonds, these, will determine the fact that DNA is double helix. Another thing to know is that the DNA strands of the single strands on both sides are equal and anti-parallel to each other, which means that if we use the new abbreviation that we studied, let's say we have one strand 5', we have guanine, thymine, cytosine and adenine, and this is the 3' end. The other strand would start from the 3' end, and it will be cytosine, adenine, guanine, thymine, and the 5' end. So as you can see, the following way is the following way that we use to abbreviate and to write down the DNA double helix. And this is the 5' end. This is the 3' end of one strand. And then, in order to complete it, because we said the nucleotides come in the same ratio to each other, the anti-parallel strand will be from the 5' end to the 3' end. The following nucleotides will form 3 hydrogen bonds between each other. This will form 2, this 3, and this 2. Overall, how many hydrogen bonds we have here? We have 10 hydrogen bonds. And this is one of the questions that can pop up in an exam. Another questions that can pop up in the admission exams is to continue and to complete the sequence of the different strand. So they will give you the strand from the 5' to the 3' end, and they will say, please complete it from the other strand. You will have to remember that the direction of the anti-parallel strand of the DNA that completes the single strand of the DNA in order to complete the double helix of the DNA starts from the 3' to the 5'. And we will see why it matters so much we have the 3' and the 5', because when it comes to translation and transcription of this sequence of nucleotides, we will see that we can only transcript and translate from one side to another and not from one side to the other one. So it's very important to understand from now. So let's sum this video up. 1. Nucleotides are the building block of both DNA and RNA. 2. Different nucleotides, guanine and cytosine, can form 3 hydrogen bonds between them. Thymine and adenine, on the other hand, can form 2 hydrogen bonds between them. The ratios between the thymine and adenine and the guanine and cytosine is 1:1 because we have double strands inside the DNA, and both of them are equal and anti-parallel to each other. They complete each other. So if you have one strand from this side, the other strand will complete it and be completely equal to the other strand, but it will be reversed, the other way around. So it will complete the first original strand. Another thing that we mention is that the hydrogen bonds between the different nucleotides have different length, different distances, and different sizes. Therefore, they give the DNA the helical shape we all know from general knowledge. And that's it for this video. Another thing I would like to add is that, as similar to the beta-pleated and alpha helix sheet, because of the different sequences of the hydrogen bonds and the nucleotides, we can get different helical shapes that we have in nature. We have the A form, which is anti-parallel. We have the B form, and the B form is the most common type we see in nature. And we have something called the Z form. Now, for the admission exam, you don't really need to remember the following names. Just know for general knowledge that the B form is the most common, and we will see and we analyze and we will work with this following form. Just for your general knowledge, know that DNA come in different shapes and sizes, and the helical is not the same all over the DNAs in different organisms and in human. And that's it. I will see you next time.